Okay, good evening, everybody. I hope I'm audible. Um, Welcome to session 9. We'll be waiting for some time for everybody to join in, and then we will begin today's session. Okay, welcome to the week 9 of the interactive sessions on genome editing and engineering, which is taken by Professor Utpal Bora of IIT Guwahati. I am Sanket from School of Biological Sciences, IIT Delhi. Today, we will be talking about the genome engineered disease models, and let's see how we have. So this time I've planned to solve questions first, and we'll take out the theory based on these questions as we have been doing earlier. This time we have more of examples in the text. So I hope you have read the content carefully. So coming to the question 1, which of the following is not a characteristic of the animal model? A close relative or closely associated with the host tissue distribution, disease progression, and similar route of infection if not identical. The disease course should be relatively shorter in the animal model to complete the efficacy test reasonably and facilitating the rapid transition to human clinical testing. The animal models have to be non-reproducible and the disease model animals should be easy to maintain, work with, readily available in adequate numbers, relatively inexpensive and free of regulatory constraints. So I'll let everybody think on this and then we'll discuss the answer to this. Okay, so the answer to this question would be the animal model have to be non-reproducible. That is incorrect. Rather, they have to be reproducible. So when we talk about disease model, it's an animal or cell that display some of the pathological processes that are observed in actual human or animal disease. Studying disease model aids understanding of how the disease develops and to test potential treatment approaches. Now, an animal model, it could be like mice, rats, zebrafish, are non-human species, but sufficiently like humans in their anatomy, physiology, or response to a pathogen that can extrapolate the results of animal model studies to better understand human physiology and disease. They are used in medical research as they can mimic aspects of a disease found in humans. By using animal models, researchers can perform experiments that would be impractical or ethically prohibited with humans. Now, talking about model organisms, numerous model organisms have been developed and are now extensively used for studying basic biology and pathophysiology of human diseases and the development of novel therapeutics. So uh, we in the lectures we have discussed the concepts in a good model design and its application, and the theory underlying biological modeling and the process of producing a valuable and relevant animal model. Development of model organisms has revolutionized our understanding of the mechanisms underlying normal development, adult homeostasis, and human disease. Many things we know about gene function in model organisms and its application to humans has come from gene knockouts, knock-ins, mutations, SNPs, etc. Now, the need for an animal disease model is that so that researchers can carry out experiments that would be impossible or impractical and ethically prohibited in humans. For example, to test the effect of drug before using it on humans, to study the safety and efficacy of periodontal treatment modalities like regenerative procedures like bone graft or surgical implants, for understanding the causes, biology, and prevention of disease. So to understand the disease mechanism, its treatment, and to overcome the limitations of human clinical trials, where you have to test um, the product or the treatment in multiple individuals, which can essentially cause certain effects which we do not want. So to prevent that, we rather screen these treatment strategies on an animal model. Now, numerous experimental animal models for diverse diseases have been successfully employed to screen new bioengineered chemical or herbal therapeutics that might have the potential for treatment of human patients. NCBI database reveals that more than 550,000 studies have been reported to use animal models for different diseases. So what are the characteristics that we aim to have as a prerequisite for a standard animal model? A close relative, a closely relative, closely associated with the host tissue distribution, disease progression, and similar route of infection, if not identical. The disease course should be relatively shorter in the animal model for completing the efficacy test in a reasonable time frame and facilitating rapid transition to human clinical testing. There should be sufficient disease correlation and pathological equivalence in the model animal, despite the genomic differences with humans. Disease model animal should be easy to maintain, work with, readily available in adequate numbers, relatively inexpensive, and free of regulatory constraints. The animal model also have to be reproducible. Okay, question two, find out which of the following is not a widely used method for generation of transgenic mice. Is it spontaneous mutation, retroviral infection, microinjection of DNA constructs, or sexually induced mutations? So talking about mice, it has a lot of advantages over other animal models. The mice genome is similar to that of humans. It has a good genetic or it acts as a good genetic or molecular toolbox. And the animal size facilitates large scale, high throughput studies, making it a cost effective model. Its potential for making medical research and in particular drug development more efficient could be increased by solving a range of identified bottlenecks. Mouse models have been successfully used to validate drug targets and to determine efficacious and safe dosage schemes for combination treatments in humans. These cases have one factor in common. They do not aim to fully model a disease or disease mechanisms, but rather set out to obtain specific functional information. Now, the most commonly used Small animal cancer models are mice, but there are several drawbacks to it as well. A typical human is 3,000 times larger than a mouse, lives 30 to 50 times longer, and therefore undergo about 105 more cell divisions in a lifetime. Mice develop cancer of mainly mesenchymal origin, such as sarcomas and lymphomas, even without genetic modifications, whereas with age, humans have a bias towards development of epithelial cancers like carcinomas. Due to the small size and short lifespan of mice, loss of certain tumor suppressor genes is insufficient to result in the development of cancer in highly penetrant manner, therefore, particularly when such mutations are heterozygous. Now, what are the widely used methods for generation of a transgenic mice? It's spontaneous mutation. So these mutations, they appear spontaneously in the mice colonies after successive breeding events and are usually detected when associated with a phenotypic change. The analysis of the genetic background of spontaneously mutated mice can be associated with events encountered in human pathologies and further used as models of specific diseases. Chemical radiation or irradiation induced mutations can be there. These are genetic modifications based on the exposure of mice to mutagens like ethyl nitrosourea, um, that can be used for large-scale programs of mutagenesis and establishment of specific genetic alteration patterns responsible for human diseases. Widely used methods for generation of transgenic mice, if we talk about the other method is retroviral infection. This method is one of the first partially controlled protocols for generation of transgenic mice and is based on the transfection of pre-implantation embryo with a retrovirus that contains the gene to be replaced or modified. The modified embryos are implanted into recipient females and analyzed for presence or absence of genetic modification in concordance with the developed phenotype, right? And the other one is microinjection of DNA constructs in this protocol. As the name suggests, it comprises direct injection of DNA constructs into one cell fertilized embryos, followed by transfer in recipient females and analysis of the presence or absence of the genetic modification in concordance with the developed phenotype. Question 3, Janish and Mins created the first ever mice with transgenes in which year? Is it 1981, 1974, 1994, or 1954? So the answer is 1974. They created the first ever transgenic mice. Although the mice could not pass the transgene to their offspring, the impact and applicability of this experiment were huge in spite of its limitation. And the word transgene was coined much later in 1981 by J.W. Gordon and F.H. Rudel, who successfully incorporated the foreign gene in the host chromosomal DNA and demonstrated the Mendelian distribution up to two succeeding generations of progeny. In 2013, the first gene knockout mice using CRISPR-Cas9 was created by Rudolf Janisch's team. Wang and co-workers injected TET1 and TET2 sgRNA, single guide RNA, with Cas9 mRNA to zygotes, resulting in production of mice that created 80% mutation in both the genes. This was the first ever demonstration of using CRISPR-Cas system for one-step generation of animals carrying mutations in multiple genes. Select the correct option about humanized animals, which are the mouse with short generation time, animals that develop from humans. They are especially transgenic animal models that mimic human responses to study. Okay, so the answer would be 1 and 3. So let's talk about it. So if we are talking about humanized animals, uh, the laboratories, therefore, they resort to developing simpler and effective models, especially transgenic humanized animal models that mimic human responses to study and understand various aspects of infectious agents, pathogenesis, disease progression, nature of protective immunity, and vaccine development. So the modification of the mouse genome, if we talk about, the mouse has a short generation time and comparatively low maintenance animal. Its production cost is also low. It's a perfect mammal for probing the genome to understand its function and its complexity. For improved understanding of human health and disease, researchers have created a wide variety of mouse models that can carry human DNA. The advances in genome engineering make possible targeted replacement of mouse genomic sequences with human orthologous DNA. This capability range from finely tuned humanization of individual nucleotides and amino acids to the incorporation of many megabases of human DNA. So this is the humanized procedure where we give sublethal dose of irradiation of in case of mice, and then we directly give them human blood so that they have similar pathology to that of humans. We perform genetic engineering, and then this is how we perform trials. So um, the number of targeted genomically humanized mouse strain is less compared to, the transgenic mouse that we are all have, right? So the humanized mouse models or humanized animal models have more relevance because they maintain the correct genomic context, right? And the gene of interest. We are not talking about homologs here. We are not talking about gene homologs in case of humanized animal systems, but rather the human gene itself. Correct spatiotemporal expression patterns. The translated protein is expected to display the unique biochemical properties of the human gene, including potentially unique deleterious properties when mutated. Where non-coding human sequences are incorporated, human-specific regulation, human gene splice isoforms may be maintained. Find out non-stem cell type from the following. Is it unipotent, pluripotent, totipotent, or multipotent? Okay, so when we're talking about stem cell, it's a kind of cell. So we're talking about cells. Uh, all our body cells, they are coming from the single source. So our body have over 200 different cell types. Each type of cell is specialized to carry out a particular function, either alone or by forming a tissue, right? The different tissues, when combined, form specific organs, are like factories where every type of cell has its own job. Same genes, yet different phenotype and function in each and every cell is observed. All these 200 different cell types within an individual human organisms are genetically identical. The variation in the cell types arise due to differential regulation and expression of genes. So stem cells are are unspecialized cells which are able to change and transform into other cell types of the body. This process is called differentiation, and it leads to the development of all types of cells in the body, right? In nature, stem cells are produced only from stem cells, but we can also artificially produce them when we call it as induced pluripotent stem cell. We'll talk about it. So stem cells are able to differentiate into any cell of an organism and have the ability to self-renew. Stem cells could be unipotent, where they could only form cells of a particular lineage. For example, hematopoietic stem cells would only form if it can only go for hematopoiesis, it can only form blood cells. Oligopotent, they can actually produce multiple cell types, usually within the same lineage. Multipotent, again, a little bit more, they are more potent, so less differentiated. Pluripotent stem cells, these are basically the embryonic stem cells, which can form all the three germ layers, and it cannot form the placenta. Whereas totipotent cells, they can also form the placenta. So these are how it is. You can read about it in the lecture slides. Okay, question 6 is the important considerations while using iPSCs as a disease model are. I don't see the slides changing. I'll just, yeah. The important considerations while using iPSC as a disease model are whether the disease is monogenic or polygenic, the penetrance of mutation, the age of onsets, or whether differentiation into an appropriate cell type is possible. Okay, so consideration for using iPSCs as disease models, we have to consider the following. Uh, Whether the disease is monogenic or polygenic, whether it is caused by one gene or more than one gene is something which is very important. The penetrance of mutation, that means how much is the mutation affecting other cells as well, whether it is only affecting one cell or the multiple cell types. That's what we are, multiple cells in particular, that's what we should be seeing. The age of onset is something, again, we should be observing, because, for example, in multiple forms of cancer, the age becomes a crucial factor. Whether differentiation into an appropriate cell type is possible, and whether there is appropriate phenotypic readout at a molecular or cellular level. So, for example, if you are creating some mutations or you're reverting some mutations back to wild type, whether phenotypically they can be observed. Those kind of things are very important. Um, so, yeah, question 7. I'm so sorry. Question 7. To induce a T11 to 22, Q24, Q12 dash are expressed in human embryonic stem cell, create cells to create double-stranded breaks in both genes. So is it ZFNs, CRISPR, talons, or meganucleases? So the Ewing sarcoma family of tumors is the second most common type of primary malignant sarcoma. ESTF includes soft tissue Ewing sarcoma, peripheral primitive neuroectodermal tumors, and Askins tumors. Ewing sarcoma is a bone-associated malignancy arising primarily in childhood and adolescence, and it's an aggressive form of cancer harboring a characteristic translocation. So this is how it happens. It from 11 to 22, there is a translocation of a particular gene element, as you can see in the figure. So there's a normal 22 chromosome and normal 11 chromosome. And from the 11th chromosome, the location Q24 it goes from Q24 to Q22. And that causes a fusion of the chromosome 22. There's an abnormal gene or chromosomes, right? So for ring finger nucleases are expressed in the human embryonic stem cell to create double-stranded breaks in both the genes. So this is how we know, and ZFN is the answer. Question 8. Talons were designed using tail NT software, and the criteria used are the following except one. Talon binding sites range from 15 to 19 bases. The spacer length was 15 to 16 bases to fill the Goldie Talon designs. When possible, Talon target sequence were selected around a restriction enzyme site, or single clones were trypsinized and transferred to 24 well plates for the selection by gene mutation analysis. So talent-mediated somatic mutagenesis in murine models was performed in 2014 by Zhang and its group. So they use genome editing tools to create and analyze targeted somatic mutation in murine models of liver cancer. And TALEN were designed against beta-catenin gene APC, two commonly mutated genes in hepatocellular carcinoma, to generate isogenic hepatocellular carcinomal lines. Both mutant cell lines exhibited evidence of WNT pathway dysregulation. Talents targeting beta-catenin promoted endogenous hepatocellular carcinoma carrying the intended gain-of-function mutation. However, talents targeting APC were not as efficient in inducing in vivo homozygous loss of function mutation. They hypothesized that hepatocyte polyploidy might be protective against talent-induced loss of heterozygosity. And indeed, APC gene editing was less efficient in tetraploid than in diploid hepatocyte. To increase efficiency, they administered adenoviral APC talents, and they could achieve a higher mutagenesis rate. Talent were designed using the tail NT software. And the criteria were that the talent binding sites were it ranged it from 15 to 19 bases. and the spacer length was 15 to 16 base pair to fit the Goldie talent design. When possible, talent target sequence is selected around a restriction enzyme site. Okay, question 9. Which of the following is not one of the most widely used methods for creating a transgenic mice? This is a repetition of the same question which we have seen earlier. So we have already talked about how transgenic mice are produced. So it could be through spontaneous mutation, it could be through chemical irradiation, it could be through retroviral infection, or it could be through microinjections. We have already talked about it. Name the first vertebrate model used to demonstrate that CRISPR-Cas9 can effectively edit the genome in vivo with up to 50% targeting efficiency. Is it zebrafish, mice, monkey, or seal? So first vertebrate model was zebrafish, used to demonstrate that CRISPR-Cas9 can efficiently edit genome in vivo with up to 50% targeting efficiency. Who created the first ever mice with transgene and when? Was it Janisch and Mintz in 1974, Janisch and Mintz in 1999, Wang and Yang in 1974, or Wang and Yang in 1999? Again, a repetition of the same question. They create Janisch and Mintz in 1974 created the first ever transgenic mice. What does the principle known as the three R's rule stands for? Is it replacement, recombination, and refinement, replacement, reduction, and recombination, replacement, reduction, and refinement, or replacement, regulation, and refinement? So the three R's, they stand for replacement, reduction, and refinement. So that's option C. It has become a standard to which every project involving the use of animals is evaluated. So whether these animal models can be replaced, whether they can be reduced in number or refined. Which of the following statement is true about pluripotent stem cells? It is characterized by the narrowest differentiation capabilities, a special property of dividing repeatedly, um, form cells of all germ layers but not extraembryonic structures, highest differentiation potential, and allows cells to form both embryonic and extraembryonic structures. So the answer to this would be it forms all germ layers but not extraembryonic structures. When we're talking about it, we mean the placenta mainly. So it cannot form the placenta. If a stem cell could form placenta, it is a totipotent cell. It's not a pluripotent cell. We have already talked about it. So that's, you can read about it. When was the first clinical trial conducted using iPSC derived retinal cells? So you can read the question. It was done in 2014. Which of the following is true regarding conditional knockout by inversion? CRISPR enhanced HDR can be used to introduce a recombinase invertible cassette. CRISPR induced double-stranded break is used to enhance homology directed repair with a 100-200 nucleotide single-stranded DNA oligonucleotide repair template to introduce small defined changes. A selectable marker cassette is introduced into an intron or non-functional region along with the SNP of interest, or catalytically dead BCAS9 is used to recruit a variety of enzymatic activities at specific sites. So what would be the option? So talking about the strategies of genome editing in case of CRISPR, Cas9, we can either go for gene knockout, right? Where CRISPR enhanced homology directed repair can be employed to replace a critical exon with a selectable drug resistant cassette, drug R, on one allele, relying on NHEJ dependent indels to disrupt the other allele. So that's how it's performed, as you can see. In gene knockout, a CRISPR induced double-stranded break can be used to effectively introduce indels. In conditional knockout by inversion, what happens is homology-directed repair can be used to introduce a recombinase invertible cassette, which is flanked by LOXP sites, um, into an artificial intron. This contains a splice acceptor followed by transcription termination signal. So in one orientation, it causes premature termination and mutation of the gene. In the opposite orientation, splicing occurs around the cassette, allowing the normal gene product to be produced in the allele. The second allele is disrupted by NHEJ-induced indels, as in option. Now, SNP introduction gene, the CRISPR-induced double-stranded break is used to enhance homology-directed repair with 100 to 200 nucleotide single-stranded DNA oligonucleotide repair templates to introduce small defined changes like single nucleotide polymorphisms. Scarless SNPs could also be performed using, selectable marker cassettes. And then there is epigenetic editing as well, where, uh, a dead Cas9 is used to recruit a variety of enzymes. Question 16, which of the following statements regarding the generation of human-induced pluripotent stem cell is true? HIPSCs are derived from embryonic cells, not from somatic cells. HIPSCs cannot be differentiated into various cell types. Somatic cells from patients can be reprogrammed into patient-specific HIPSCs, or differentiated HIPSCs can only be used in cardiovascular applications. So the answer to this is somatic cells from patient can be reprogrammed into patient-specific iPSCs. So as you can see in the diagram here, it shows a summary where somatic cells are harvested from the patient and reprogrammed into patient-specific iPSCs. They can be differentiated into multiple cell types such as cardiomyocytes, smooth muscle cells, endothelial cells, which can be used for different applications for disease modeling, drug development screening, regenerative medicine, or tissue engineering. Question 17. Which combination of transfected elements should be used to correct the common G691A mutation in the IL2RG gene, exon 5, which causes XKID, excellent SCID, that is severe combined immunodeficiency in pluripotent stem cell using homologous recombination mediated TALEN. So do we need to have TALEN expression plasmids and a therapeutic donor of cDNA other than exon 5? Only TALEN expression plasmids, TALEN expression plasmid therapeutic DNA of donor cDNA of exon 5, or only a therapeutic donor cDNA of exon 5. So transfection should be done using TALEN expression plasmid and the therapeutic donor cDNA of exon 5, both of them. Which of the following is used as alternative to embryonic stem cells in precision regenerative medicine? Is it mouse cell, rats organs, human organs, or iPSCs? So obviously the answer is iPSCs in this case, right? It's obviously iPSCs because it will have all the genomic changes that are particular to that infected or the patient rather. So hence it gives us the most flexibility which is required for um, precision medicine. So with that, I think we are at the end of this session. If you have any questions, now is the time to ask. If there are none, we may call it a session and end the session right here. I'll stop the recording.